A very warm afternoon to all of you, and once again, really thank you for joining this Young Investigators Forum organized by the Tanzania Society of Human Genetics. Today, my topic will be on the changing the landscape of genetic healthcare in Tanzania. I think Siana has made my work very easy in terms of really starting the conversation as to why we really need genetics in the medical field and what is so important about it and where are we heading. So I hope by the end of this presentation, you will all be able to take back the message as to why is genetics so important, what has been done in the continent, where can genetics be applied in the medical field, and what is the future in terms of Africa and Tanzania. So once again, my name is Mohamed Zahir Ali Mohamed. I am also the co-founder and general secretary of the TSHG. I am a co-investigator at the Sickle Cell Program. I'm also a lecturer at the Department of Hematology at Muhimbili University. So in this presentation, I will go through a couple of things. First, I will very briefly introduce what human genetics is all about, how we can implement this genetics in the medical diagnostics world, what exactly is Human Genome Project, why is it so important, also genetic variation, or if you want to call it genetic mutation in the African population, why it is so important. What is the current issue as far as the problems that are associated with colored populations within the Human Genome Project and other researches that are happening around the world. But not only that, I will also talk about what are the initiatives that have been taken by African geneticists and Tanzanian geneticists to cater those problems that came about for colored populations. But again, of course, we also have pressing issues in Africa that we must address, and we will talk about that towards the end. And also the reason why we formed the Tanzania Society of Human Genetics and what is to come. So just to sort of start the talk, I'm sure everybody knows what this is. This is a normal cell. Within a cell, if you leave aside the cytoplasm and you really focus in the center, so I don't know if you can see my mouse. So really this center area, this is the nucleus. And we call it the central dogma of life when we produce proteins by the cells. And we have to ask ourselves how these proteins came about to being. It all starts with this code of life called the deoxyribonucleic acid, the DNA. And the DNA then undergoes something called the transcription, which then produces the mRNA. And the mRNA then undergoes translation process to produce the protein. This in itself is called the central dogma of life. Once you have understood this concept, you will now be able to understand why understanding this concept is so important. So let's see how this is relevant for genetics. Within our cells, we've got a total of 46 chromosomes, which means we have about 23 chromosomal pairs in each cell. Now, 23 chromosomes we get from our mother, and 23 chromosomes we get from our father. So what you are seeing here in this slide is a picture of a karyotype, whereby the chromosomes that we get, so a pair from the mother, a pair from the father, have been organized in groups of 22 pairs. And of course, if you are a female, you will have a double X chromosome. And if you are a male, you will have one X chromosome and one Y chromosome. So this is essentially when you stretch the DNA and compact it in a way that can be fit in a cell. Just really basic biology. Now, at the Wellcome Collection, which is one of the Wellcome Trust Centers in the UK, they decided that they wanted to write down the entire code. You see, the DNA is made up of 4 building blocks, the A, T, G, and C, which I think you can see from here. So if we take the A, T, G's, and C's in our DNA, we can essentially fill up the entire row of information here that you see in the picture on the left. So there are about 3.4 billion units of DNA code, and basically this is 127 volumes of the book, with about 1,000 pages per volume per book. So you can imagine how big the amount of DNA information, or the code in itself of the DNA, is extensive. So some fun facts to just throw around. If I was to sequence any one of you, and supposedly, let's say I have a Microsoft Word document and I would simply type ATGC, the size of the Microsoft Word would have to be 200 GB for me to have actually completed the entire code. So you can understand how massive the amount of data every human being is actually carrying. But then what is also very interesting is that 99.9% of the DNA between all of us in this group, in this meeting today, is very similar. It's that 0.1% of the DNA that makes me, me, and you. And that is also within that 0.1%, we want to understand what are the predispositions or challenges that lead to disease, that leads to lots of other conditions that the normal person can be distinguished from the diseased person. And if I was to take your DNA from your cells and lay it out in a line, a single line, I would be able to, with the length of the DNA, I would be able to go to the sun from the earth and come back 70 times. So I'm just trying to tell you these kinds of information so that you are able to understand how much information we all are carrying and how relevant this is then going to sound when it comes to looking for those mutations that cause diseases. So when we talk about mutations or variations in the DNA, we have a large number of types. So the first region that you see in this slide is called the single nucleotide polymorphism. Many times these are called SNPs. And basically you have your reference DNA sequence. So here within the dark gray shaded box, you have a C, but now the C turned into the A in the same position. And this can very well be the case that leads to disease. So from those 3 billion nucleotides that make up your DNA, it would probably just take one change from a C to A to cause a disease. And this is how diseases like the sickle cell disease occurs, or the thalassemia occurs, and various other diseases that are monogenic in nature. We also have different other variations, for example, the simple sequence repeats. So instead of, you see here the CA, 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 you have an additional CA towards the end of the sequence. And that can also create a problem. We also have something called the insertion deletion polymorphism, also called as indels. In the top, so assuming all the top rows are the reference and all the bottom rows are the patients. So at the bottom, you see the 3 nucleotides are missing. So we call that an indel. And we also have block substitution, inversion variant, copy number variations, segmental duplication. So this is your chromosome that I showed you from the earlier slide, whereby you have a duplication of the segment, or a translocation, whereby a piece of chromosome 1 will go and sit right behind a piece of chromosome 6. And so then now the patient is carrying up 1 and a 6 in the same cell. And that can also become disease causing. So these are the types of variations that can occur in the DNA that leads to diseases. Now, how do researchers compare genomic information to identify genetic alterations? You see on the left, those that are in red have a condition. And those that don't have a condition, sorry. Those in green on the right are people with a condition. If we compare DNA from people without a condition and the DNA from people with a condition, we are able to understand what is different between those sequences. And the differences in those sequences will then tell us if those sequences are in fact causative of that disease condition. And this is basically what researchers do with that genomic information, simply to compare the information and find out if that's actually causing a disease or not. Now, how you probably want to ask, how can we implement genetics for medical diagnostics? So I'll give you a short overview of how this is used in the West. and how they sort of streamline it for their hospital usage. So assume that you have a patient. The patient comes to the physician. She complains of conditions, and the physician says, well, this could be something that can be caused by a mutation in your DNA. So she requests the patient, let's get your genome sequenced so that we can find out if there is a mutation in your DNA and if something you have inherited from your parents. Then the patient undergoes a consultation. So there is a genetic test consultation service whereby she sits down with a genetic counselor, a molecular pathologist, a medical geneticist to understand, based on those disease conditions, which genes would be ideal to screen. And following that, there is a multidisciplinary team, for example, that includes a genetic counselor, a treatment team, a biocurator, or an expert, to establish questions as to why the patient is undergoing certain conditions. After, of course, and I hope in the future the NHIF will cover this, but then afterwards, once you have the genetic test done, the insurance will give a go-ahead as to the cost of the sequencing. The patient meets with the counselor, and then the consent forms are signed, and you meet a nurse where the blood is drawn. And once the blood is drawn, the DNA is extracted from the blood, and it is subjected to a machine like this on the left. It's called the Illumina genome sequencing machine, whereby the ATGC code of the DNA is recorded. And once the ATGC code is recorded, the data is then aligned. Then we understand what kind of variant is there at a specific location. Of course, we also want to check the quality of the DNA because we want to be absolutely sure that the mutation that is going to be called by the machine is in fact true. And then we go through something called variant filtering and prioritization, because essentially you have to understand that when a patient is carrying a mutation, a lot of other people are also carrying mutations. So it is not necessary that when you see a SNP, then that SNP is causative of the disease. It only has to be causative of the disease when it is a biologically relevant or actionable, or when we know it is really so rare that only with a specific subset of patients which have the same disease are carrying the mutations. So it has to undergo a number of variants filtering. So this is another process in the workflow. And then the variant verification gets certified by the lab, and then a report is made. And then from the report, it goes back to the medical doctors who are from a specific field. So if you have something to do with cancer, then you'd go to an oncologist. If you have a problem with your heart, you would go to a cardiovascular specialist, or if it's a child, you will go to a pediatrician. So that is part of the return of data processing. And from there on, the patient then goes and meets back with the genetic counselor to see how then the treatment can be done based on the findings of the results. So this is sort of an overview of how the workflow or the genetic testing can actually be put into practice in the medical world. Now, the Human Genome Project. So Human Genome Project was the idea behind making this Human Genome Project is really to understand what is the reference sequence. Because if I am going to tell that this particular mutation is causative of a disease, I need to have the DNA sequence of a normal person to say that this is where the difference was, and that difference is causing the disease. So the idea of having the entire 3 billion blocks of human DNA as a reference, so that we can use that reference for comparisons, is basically the whole idea of what the Human Genome Project is all about. And this guy down here on the right, his name is Francis Collins, and he, and together with his colleague Craig Venter, were the first ones to actually conceptualize the idea of really performing the Human Genome Project. And so the project started off in 1990, and it ended in 2003. And they established a lot in the process, lots of milestones. For example, they managed to sequence the first bacterial genome in 95, the first yeast genome in 96, a mouse genome in 97, the E. coli genome in 99, and the roundworms were also sequenced. So within that process of human genome sequencing, we were also sequencing species that we live around us. So the goals of the Human Genome Project was really to determine the entire sequence of the DNA, identify the genes in human DNA, store the information in databases, improve the tools for analysis, and transfer the related technologies to the private sector, as well as also discuss the ethical, legal, and social issues, which I believe will be touched upon during our session tomorrow. So I will not address the ALSI today. Now, what has happened over the process? See, the Human Genome Project, when it started off, it took $3 billion to complete, which is terribly expensive. But because of the improvement in the technologies, the prices of sequencing people went down. So in 2005 and 6, the price went almost half of what they started off with. In 2008, it went tenfold down. So for every genome, it costed 100,000. In 2010, it became less than 10,000. 2015, it became less than 5,000. So basically, and today we can sequence a genome with as low as 800 euros or $1,000. So the innovation in chemistry, fluidics, computational hardware, bioinformatics has allowed us to get more data at a cheaper rate. And so obviously, if we are now able to get more data for a cheaper price, you would expect a more number of genetic tests that will be available over the years. So what started off as something below 500 in 1993, in 2013, we are looking at something between 2,000 and 2,500. Today, if we look at the number of genetic test consultations that are happening around the world, I'm sure this is even 100-fold more than what we saw in 2013. So basically, this is how the trends of the genetic tests are happening throughout the world. Now, this is a picture that was taken in 2006. Basically, each line you see is a picture of a chromosome. So these are all chromosomes in our DNA, which I've showed you a picture of. And for example, in this one, this is the first chromosome. Every dot that you see here represents an area whereby a mutation has been found to cause diseases. Now you can see in 2006, not that many dots can be seen, because at that time, we still didn't know a lot more about our DNA. But what happens slowly. So in 2007, in the second quarter, more number of dots are increasing at various locations throughout the genome. In 2008, more dots, and see how fast the number of mutations that were found to cause diseases has sort of increasing. In 2009, even more. This is a picture that was taken in 2019, and you can clearly see that, in fact, in all the chromosomes that are there in our DNA, there is a location in which a mutation is found to carry a particular mutation for causing a particular disease. And you can see a wide variety of diseases that are included. So there's a digestive system disease, cardiovascular disease, an immune system disease, hematological diseases, responses to drug, cancer, and so on and so forth that are all causative or associated to be causing a disease. And all this thanks to the technology that we currently possess. Now, the kind of information one can get from the DNA, it's not all necessarily that if there's a mutation, it will cause a disease. No, sometimes a mutation is there and it is a silent mutation. Basically, the mutation doesn't do anything, but the mutation does exist. Or there is a variation which can tell you what eye color you're going to get. And that's not necessarily causing a disease. It simply tells you that that mutation causes an eye color. Or some people are tall, some people are short. Some people have gray hair, some people have black hair. And so all these things can be defined by the mutations we carry. And not necessarily all mutations cause diseases. We also have mutations that talk about differences in response to the diet. For example, obesity, or the effect of antidepressants on what a person takes, or the influence of likelihood of a disease, for example, diabetes or celiac disease. And then some directly result to a genetic condition, whereby, for example, if you have a mutation on the HBV gene, then you can get sickle cell disease, and so on and so forth. And so with information like that, scientists can use that mutation information from a patient to create drugs that are more specific to that patient. So in this picture, you can see that when there is a normal gene, you have a protein that interacts with the drug, So it sort of fits, the protein and the drug fit together to cause the desired effect. But now when you have a mutation or an altered gene, the protein produced, remember the central dogma of life, that the DNA code dictates what protein will be produced. And so now when you have an altered protein, the drug will not fit. And so what happens is that if you have this information, then you can modify the drug so that it fits with the altered protein that is being carrying by the patient. Now let's come to the African population. Let's come to the genetic variation that is present in the African population. All the information that we have so far, there was a huge study that was done. It's called the International Haplotype Project, or the International Consortium of Human Genomics Projects. And what they basically did was they assembled a number of people from all over the world, USA, Africa, Asia, Australia, South America, and they sequenced the DNAs of the people from those areas. And what they find out are two very important findings. One is that the amount of mutations that Africans possess was far more, as you can see here in the graph below, was far more than the populations in the rest of the world. And two, that the African populations had very diverse information within their DNA. So not only are we carrying more mutations, not necessarily all our diseases, but we are carrying more mutations, but we are also so diverse from each other. So a population in Tanzania will be very different from Zimbabwe or South Africa, whereas a population from Denmark and Norway probably are very similar. So there is that difference also to take into account when we are making decisions about medical diagnosis, or whether we are making decisions about therapy, because that information is very specific to a population. Now, why did this happen? So this is a picture on the A column here. These are 10, 11, and 12 are 3 patients. And these are the DNAs of all the people from different countries and how they match with it. You can clearly see that the DNA from people that are coming from West, Central, and South Africa are all looking similar. And people coming from Europe are all similar. But within Africa, the South Africa looks so different from the Horn of Africa, which is the Somali area, which is different from North Africa, which is different from Western Asia, which is different from Southern Asia. My point is that, you know, there is so much difference within the African populations themselves, whereas there is not so much difference of DNA between European populations. Why? And this has all to do with the human migration. You see, at some point, everybody in this world lived in Africa. We are all children of Africa. Africa is considered as the cradle of mankind. But when the human migration happened, which occurred in 2 routes, one was through the Horn of Africa, so the Somali area, as you can see here in the picture on the left, and one from the lower part of Egypt, as you can see from here. So about 300,000 years ago, these people migrated out of Africa via those routes. And what happened, it's like if you have experience watching the Days of Our Lives show, you know that clock that has sand in between, and it sort of goes from one area to the bottom. So it works in the same way, whereby population within the continent are filled with multicolor. But because of the bottleneck of the migration pattern outside of Africa, there are only few colors that manage to come out. And when those, so supposedly you have 12 colors in Africa, only 2 colors manage to come out of Africa. Now, what happens is that these 2 colors that came out are also intermarriages happening. And so the children that they will produce will also be one of those 2 colors. Eventually, the entire population of Europe and other countries will be of the same color. And this color now represents the genetic variation of the DNA, which is why there is so much difference between African populations and the rest of the world. And it all has to do with the human migration. In another paper, they also showed this, that modern human origins happened about 200 to 50,000 years ago. And you can see in the left part of this slide that the North African and African populations have a lot more variation as opposed to the Eurasia, Asia, and Oceania. So I won't go into specific details, but I just want to show you this picture to show you that the further away you are from Africa, the more likely it is that your DNA will be different from Africans. And this is basically showing how the whole migration pattern has left an effect on the variation on the human DNA sequence. So when I go back to the Human Genome Project, I want to touch upon the idea of the underrepresentation of colored population. You see, when the Human Genome Project occurred, lots of people were sequenced, and lots of genetic researches are happening around the world on diseases. But the problem is that the researches that are happening are all happening on populations outside of Africa, on populations that are Caucasian, that are white, that are European. Not much is given importance, or not many African populations are put in that basket of the DNA sequences so that more Africans will benefit on the diseases. So if I'm going to use information about genetic diagnosis of, let's say, a cardiac condition by using the data of a European population, I'm afraid as a medical geneticist, I'm not going to be able to diagnose patients that are residing in Africa because they have a difference of the genetic DNA. And therefore, that has also consequences to the therapy, because now those drugs that are most suited to the European population is not necessarily going to work for the African population. So this created a lot of, uproar on genetic social media, in the news, that why is the genetic diversity not so, why African populations are underrepresented, but we need to do something about this. There's a huge problem with the Human Genome Project. And there were lots of studies that happened. Now, this guy, this man here, his name is Professor Charles Rotimi. He's a Nigerian, and he went to the NIH in the US and was among the first ones to look at the African genome. And what he decided to do was to found the Africa Society of Human Genetics, as well as the H3 Africa, which today are model institutions for improving genetic research on the populations of Africa throughout the world. And what he basically decided to say was that, you know what, Black genes matter. Black lives matter. This is something that we are hearing today, but Black genes matter already was something that was going on quite a number of years ago. So the initiatives by African geneticists include the formation of the Africa Society, and within the Africa Society, formation of different national societies. So we had the Egyptian, Mali, Senegal, Cameroon, Rwanda, Congo, South Africa, and various other institutions that took up research that was funded by the H3 Africa. And in 2018, so these are all the members that participated in the opening of the Africa Society of Human Genetics and H3 Africa, including the person who was initiating the Human Genome Project, Francis Collins, right together with Charles Rotimi in this picture. So when these uproars happened, now more universities within Africa decided to take up genetics, and more African DNA started being sequenced. And so that we have understood now more better that African genetics is very diverse, and there is more that is needed to be done to unlock the disease susceptibility. But also, there are challenges and disparities in the application of the personalized genomic medicine, for example, for people with African ancestry. And lots of papers started coming out about the genetics of Africa, where they come from, the migration pattern. But of course, this is not enough. Africa is built with 54 different countries with 1.3 billion people. And we still have a long way to go before the entire continent is equally or adequately represented in the genetic databases around the world. So, therefore, my take-home message to you is that Africa's people must be able to write their own genomics agenda. It is not necessary that a white man or a European should tell us what we should be doing. I think it is important that we should be dictating what we should be doing based on what we see is needed for our continent. And just to give you an overview of the latest researches that has happened, is that in the left-hand side, there was a study that was done that looked at sequencing of 900 African people, and they found that almost 10% of the DNA that was found here, 10% of the DNA that was found from the results of the study never was captured in the Human Genome Project. So imagine a whole chunk or I should say 10% of 3 billion nucleotide base pairs was found from this study. And secondly, in this study on the right, this was a more recent paper released in October 2020, that they were, when they looked at 50 ethno-linguistic groups, about 400 people that were sequenced, they were able to find 3 million new undescribed variants that were found. So as more and more African genomes are being researched, more and more information we are now beginning to understand that really the Human Genome Project and other research databases were not able to capture. And hopefully this will be the start of various other such initiatives so that we can improve how we diagnose our patients and how we provide treatment for them. So some of the pressing issues, of course, that exist in Africa is really building research capacity for genomics research, improving medical genetics services, setting up these genetic testing laboratories, cytogenomic techniques. For example, the first picture I showed on the karyotype of those pictures of the chromosomes. So those are also part of the cytogenomic techniques, which should be used as a diagnostic tool on a routine basis. Ethical considerations in genomics for Africa, biobanking, whereby we can store our samples, biorepositories, and also bioinformatic training challenges. We have a lot of ground to cover in terms of the kinds of issues that we have in Africa. Also pharmacogenomics in Africa and various other issues which are still at the moment limited. But I think together, coming with the societies, and one among them is our society, the Tanzania Society, we will be able to tackle some of these issues slowly and steadily. So my dream as an individual is to be able to go to a hospital somewhere in Tanzania, and the first thing the doctor, and if it's a genetic condition, then the first thing the doctor will ask me is for a family history. He will counsel me on the genetic condition. There is going to be an infrastructure for sequencing, variant calling, and the insurance will cover it so that I have access to clinical care, particularly for my genetic condition. And together with that, we also have to understand something called the precision medicine or personalized medicine. We have a lot of different groups of people in between in the picture, but we cannot treat all conditions with the same drug. So we have to understand the DNA of these people to determine which drug will be beneficial or which drug will not be beneficial to a particular population of people. So that I don't want to give prescription to a person who I know for sure the drug will not benefit. And I don't want to lose out in giving a prescription to a person who I know will benefit. So these are some of the dreams that I really want to see happen in Tanzania. So with that, I just want to once again invite everybody for the 13th conference of the Africa Society of Human Genetics and the first conference of the Tanzania Society of Human Genetics, which will happen next year, 2021, from the 30th of August to 5th of September. And the theme of the conference will be Genomics and Translational Research to Improve Health in Africa. This is the first time that conference that initially started in 2003 will now for the first time come to Tanzania. So all of you, please note down these dates, and I really hope all of you will be able to attend this in Dar es Salaam. So with that, I thank you for your time, and I'm happy to take any questions. Asanteni sana.
Thank you, Mohamed. Let's see, I'm not sure if I've seen any questions in the chat. Does anybody has a question? You can either raise a hand or type in the chat if you have a question, a comment, anything. No? Okay, I don't see anything in the chat. So while you're thinking about questions or comments, oh, there's a question from Zainab. Karib Zainab, I think you can unmute the mic. Please go ahead.
Hello, can you hear me?
Yes.
Thank you very much for this wonderful presentation and this really good symposium. My question is that it's really nice to hear that there is people now thinking about African genetics and how we can be more represented in the Human Genome Project. So as a healthcare professional, what would you say is my role on a day-to-day basis? What can I do in my own capacity to ensure that we get this equal representation? Because in our own capacities, not everyone will become scientists and will be able to do, hands-on research in terms of uh, getting the DNA sequence. But in our own capacities as healthcare professionals, what would be your advice? What can we do so that we can also contribute to this society in the long term?
Thank you. Mohammed, would you like to take that
Yes.
first?
Thank you very much for that great question. And I think that is a very, very relevant question, because you don't have to be necessarily a geneticist to really be able to be influential in driving the genomics agenda forward. For example, let's take an example of a pharmacist. Today, we know pharmacogenomics is such an important thing. Simply by understanding what kind of drugs are, let's say, labeled with a pharmacogenomics criteria, and understanding how much consumption of those drugs take place in the country would be a step in the right direction to understand, in terms of changing the policy, that why are we taking a specific drug that is pharmacogenomic labeled but not clinically tested? Or furthermore, even for example, for nurses, how can you identify a person with, let's say, a dysmorphology, or that is perhaps a hereditary condition? And that would be the role of for example, a nurse to do counseling, or to be able to identify the condition, or to a medical doctor who will be able to identify those patients. So my point of saying these things is that, of course, genetics is the central piece, but it is not the only thing. We need even lawyers in this particular field. For example, those that are crossing the boundaries by crossing the ethical guidelines on how to use DNA. And that's where we need the lawyers to work in and understand how genetics works, so that they can fight for our cases. We need ethicists to understand and be able to guide us on how best ethically we can drive the agenda forward. We need nurses, we need pharmacists, we need medical doctors, we need geneticists. And on top of that, we need even computer engineers. Because end of the day, remember, the amount of DNA that we are generating is going to be analyzed on a piece of computer. And so we need a person who is able to code, a person who understands how computers work. And based on that knowledge, we'll be able to identify areas where we think that mutation is located. So I think this field is a multi-dimensional field, which has potential to grow on various fronts, whether it is from an ethical legal perspective, whether it is from a pharmacy perspective, by really looking at which drugs would be useful, whether it is by simply identification of the patients, whether that's medical doctors or nurses, whether that's from the laboratory perspective, so really the laboratory scientists who are going to be the driving force behind the isolation of the blood, whether it is the genetic counselors that are going to be able to come and talk to the patients to make them understand what the diseases is all about. So in that note, I think the entire health field needs to come together, and everybody, in my opinion, has a small role to play, but particularly with starting individually, what is genetics and what your particular field can do. I hope I managed to answer your question.
Thank you, Mohammed. I hope the question is answered. So there is another question from Mwabia. She asked, what do you think are the barriers for Africa to achieve such heights? Can you respond to that briefly?
Yes. So one of the challenges that we have is, of course, funding, is the finances that don't allow us to put the necessary infrastructure in place. And that sort of slows down the process. It really becomes a hindrance. The second thing is we don't have enough courses. So really from the training perspective, we don't have enough universities that teach genetics, and therefore, that also is part of the awareness creation. Because if you have more and more people that are being taught genetics, then more and more people will know what genetics is all about and further it. So those are some of the challenges, really training perspective, but also, for example, the government. We don't have specific, let's say, budgets that go to the non-communicable disease, which is now beginning to change. But there are some things that still need to be worked on. And those, I believe, are some of the barriers.
Thank you. I hope that sheds some light on what can we already start to work on. Another question relates to the annual meeting. I'm not sure the annual meeting, I think it's the 15th conference of the African Society of Human Genetics. Monica asks whether the meeting will require a registration fee. I think there will be a registration fee which has not been set yet. So this will be communicated as time approaches. And also usually we provide sponsorship to early career researchers. So also look out for that information. So Grantina says, thank you, Mohamed, for a great presentation. And also Rehema says, great presentation, well understood. Glorian has a question. Glorian asks, in the point of diagnosing, will it include diseases which are inherited or other communicable or infectious diseases? Mohamed.
Yes, so I think genetics is very, very broad. It is not only touching the idea of inherited diseases. I think genetics is also now becoming relevant because you see, when there is a disease, the disease is not necessarily entirely down to the DNA. There is a lot of factors that come into contact between the environment and the relation of the environment with the DNA. So more and more studies are now also trying to understand why or how the environment has an influence on the disease prognosis. And that has also come into effect in communicable and infectious diseases, such as HIV, TB, and other diseases. So the environment, also the microbiome, so really the bacteria that live in the gut and how that has an influence on the disease progression, or the geographical location that a person lives in, how that has an influence on the prognosis of the disease. And so it's really a multi field, I would say, a multi-field correlation between the environment and the genetics. I hope I managed to answer that.
I hope so. So there are a few more questions. I'll just read them out, but I request Mohamed to continue engaging these questions in the chat section so everybody can see while we move to the next presentation to try to finish on time. So I'll just read the first question from Diocles. Diocles says, thank you, Mohamed, for the wonderful presentation. My question is, do you think Africa is ready in terms of funding and well-equipped personnel to run all these projects? If not, what should be done to make this possible? Mohamed will take that. And the question is, in terms of increasing funding, how can we lobby perhaps to the government or other organizations to increase funding and therefore awareness and findings? So Mohamed, also Eneza raised his or her hand. So Eneza, can you also type in the chat box? And Mohamed will respond in the chat box to the question.
Can I just quickly answer these two questions, because I think they're really connected, and I will take less than a minute.
Briefly, we're running out of time.
Yes,
Briefly.
yes, I'm sorry. So really about funding and lobbying, that is really, really important. And one of the components of TSG is also advocacy. So for us, the responsibility is to create awareness, to engage with the patients, to really, provide this information on the social media. And therefore, when more people are aware of what genetics is all about and what genetic diseases means, then there will be a bigger voice that together will go to the government, which then brings about changes. So my only view is, or my only advice is, let's work together to really shout on social media platforms as to what genetics is all about and what genetic diseases are, where our patients are, so that the government will listen. And then hopefully that will change how funding comes. Thanks.